Hi everyone, welcome to the HIV Cure podcast with me, Sakabo Sisala Marumo, where we bring you HIV cure information in simple language. And I am just a young person who's openly living with HIV, and I just come here to advocate for an HIV cure and also just to explain and empower people of what is happening around the world in terms of HIV cure, how far we are with it, how close we are with it, and also to just break the myths and misconceptions that are there. And of course, I have exciting guests lined up for the entire season. And today we're back again with our favorite doctor. And yes, I keep bringing the young doctors because this is a youthful show, and also we need to bring the voices of young people. Today we're talking about the science of HIV cure research. On our previous episode, on episode 1, we talked towards the end of the episode. There were a lot of questions that we talked about that need to be answered, and today we're diving a little bit more on that. And in this episode, we're going to provide a closer look at how barriers to curing HIV, as well as some of the strategies that researchers use to overcome them. Hi.
Hi, Sakabo. Thanks for having me again.
Thank you for coming back. You explain concepts in such a nice way, and that's why we keep bringing you back, so that we get more information. And also from our last episode, we could hear that you still have so much information, but the time was just limited.
Yeah, this is something that you can definitely do a deep dive into, and we can talk about this for ages.
Can you please just introduce yourself to our listeners and to those who are joining us for the first time?
Sure. My name is Dr. Sherazaan Ismail. I'm a senior scientist at the International Center for Genetic Engineering and Biotechnology at the offices based in Cape Town, South Africa. I'm an HIV cure researcher, and I've been doing cure research for, I think, about 8 years now. And I started in my PhD. So yeah, I've been delving into a little bit of this cure thing for a bit.
Okay, well, yes. So you can hear that she has experience. She has, all the information that needs to be shared with you, with the young people. And, today we're talking about the science of HIV cure research. And just to dive into that, we know that when it comes to cure research, there are a lot of challenges that are there due to how the HIV virus has the ability to just hide and persist in the body, like you mentioned on our previous episodes. And some of these challenges, like you did say, that we have HIV reservoirs as well, reservoirs and latency and immune evasions. But I am so happy that I am not the one who's going to be explaining these words and these processes. And just to dive right into it, please explain to us what HIV reservoirs are and why are they considered to be the biggest challenge when it comes to curing HIV?
Sure. So we all know that antiretroviral therapy does not cure HIV. And this is because HIV integrates into our human DNA. So basically, it makes a little cut in our DNA and just pastes itself in there, and then it can do its thing. So produce more of itself. Antiretroviral therapy effectively blocks this replication, but it's not able to cut out the HIV or remove it from our body. So it just hangs around there until either the antiretroviral therapy is unable to block it anymore. So let's say a person living with HIV who is suppressed on therapy, that they stop their treatment, the virus can now start replicating again. And basically, this pool of viruses that's integrated into our DNA that we're unable to remove is called an HIV reservoir. And that's what's considered the biggest challenge to curing HIV is these reservoirs just hang around there until they are either able to produce more of themselves or the immune system clears them. The other barrier to clearing this reservoir is that these cells are also able to hide from the immune system, but we can chat about that a little bit later. In essence, it's just very, very difficult to get rid of these cells that contain HIV DNA in them because they're difficult to be identified. Essentially, HIV is using those cells to hide from the immune system. And yeah, that's why these reservoirs are the biggest challenge in curing HIV.
Okay, so it sounds like HIV is a very smart virus, and it's sort of like found its way to make sure that it lives on forever. And now I want to know what HIV latency is and how does it allow the virus to hide from these treatments that people living with HIV are taking, as well as the immune system, because you did explain on our last episode that the body has CD4 cells, and it's that cells that are supposed to be protecting the body, but we see that with HIV it's able to be there, even though it's a foreign body with CD4 cells there, it's still able to be there in the presence of these CD4 cells.
Right. So HIV latency is just the state of the virus being dormant in cells. That's all that HIV latency refers to. And so HIV is not the only virus that can go into a latent phase. There are other viruses that can do that, both in humans, in plants, it's a common strategy amongst viruses to survive, in the face of some kind of immunity or, protective mechanism that the individual has to get rid of this virus. And so, yeah, the HIV reservoir is typically viruses that are in latency. Latency forms due to a number of reasons. So, it is in part because of the antiretroviral therapy suppressing HIV replication. That means that it can't do much other than just hang out in the cells. It's also the fact that HIV is able to infect CD4 T cells. So these cells, your body doesn't want to get rid of all of these cells because they are the cells that protect your body. So the fact that HIV is able to enter CD4 T cells and become latent in these cells protects them further. You understand that your body would not want to attack its own cells that are protecting it. So it takes advantage of that fact. Not intentionally, but it unintentionally just ended up that way.
Okay. Okay, and then you also, you're talking about how HIV is able to integrate in a person's DNA. So then how does this ability make it very hard to cure HIV as compared to other viruses? Because you did mention that it's not just HIV that has the ability to go into or infect cells. We have other viruses as well that are able to do this. So why is HIV then difficult to cure?
Right. So it's also difficult to cure the other viruses that are capable of doing this. I think anything that integrates into our DNA just automatically makes it harder to cure because of the fact that you are targeting DNA that's in our cells. So first of all, how do we get something to target those cells specifically? Only the cells with the virus in it. It's not every single cell in your body that contains the virus, right? And so getting a treatment specifically to those cells, that's one difficulty. The second one is if you consider how HIV just like a pair of scissors cuts itself into our DNA, how do we find something that can cut HIV out of it without targeting our own DNA, like the stuff that's important to help our bodies function. And those are some of the difficulties that we've encountered with certain treatments. So I don't know if you've heard of CRISPR-Cas9, that can be used almost like molecular scissors that can chop up DNA and take a piece of that DNA out and can be very specific. But even with something that's that specific, we get what is called off-target effects. So there is some targeting of the human DNA that can be troublesome. And so this is just some of the difficulty with targeting it. Also, when cells become latent, there's no way really to identify them as infected with HIV. And so it's very difficult for us, but even our immune systems, that's why our immune systems don't clear cells that have latent HIV in them, because basically there's no way for our immune systems to identify those cells as having HIV in them. So yeah, that's just some of the roadblocks that we have encountered with curing HIV.
Okay, thank you for just explaining that. And a lot of people, when you just ask them around why it's difficult to cure HIV, a lot of people are informed, or they know that it's because HIV mutates a lot. Like, a lot of people know this reason so much. And now, please just explain to us how or why is HIV so effective when it comes to mutating, and how does this then impact the development of an HIV cure?
Right. So, HIV is really, really good at mutating, as you say. It's one of the best viruses that, yeah, it's really good at doing that. And so how it does this is that it's got, if you think about, okay, let's take a step back. If you think about how our DNA replicates itself, or how viruses replicate themselves, they use little proteins to do this, and you can think of them as like little typewriters, right? So they read the code, and then they retype the code, and then that's basically a copy of your DNA, your genetic code. And HIV's little typewriter is not the most effective at correcting its mistakes. So it just makes a whole bunch of mistakes, like a faulty typewriter. And so, oh, maybe I'm using the wrong analogy. People don't know what typewriters are anymore. Like a faulty keyboard. Like, if you have a faulty keyboard and it's just like typing gibberish, even though you're trying to type words, this is how HIV's little copying machine works, right? And so by introducing all of these mistakes, it introduces mutations. And some of these mutations are detrimental, so it leads to viruses that are non-functional. But because the virus replicates to such a big amount, so one copy in one cell can lead to more than 1 billion copies of HIV being released from that cell. Can you imagine if there was only one mistake per copy? That means that there's more than 1 billion different new little HIVs that are slightly different little HIVs that are bursting out of that cell. And so as the virus replicates cell after cell, these little mistakes accumulate, and we refer to these mistakes as mutations. And so mutations can either be neutral, have no effect on how the virus functions. It can be detrimental, so it can result in a non-functional virus. And in, just another, it can become more functional. And so either better at infecting, or better at hiding, or better at evading the immune system. And so, essentially within one individual, you have all this diversity of viruses that develops over time as the virus is replicating. And that's also what makes it difficult to target, because you're essentially targeting slightly different viruses all the time. And that diversity becomes even bigger when you look between individuals. Which makes achieving a global cure even more difficult, because we have to account for all of this diversity that's caused by the mutation. So it doesn't only impact a cure, it can impact preventative treatment as well. And so ARVs can overcome this because they're not specific to a sequence of the virus. They work along the viral life cycle. We know some of these mutations can also make the viruses a little bit more resistant to the treatment, or a lot resistant to the treatment. But for the most part, if you use these antiretrovirals in combination, then the mutations have no way of escaping the drug. That's why we use combination ART to overcome the mutations. And so, yeah, HIV's effectiveness at mutating makes it quite a bit of a struggle to deal with. Hence, the development of a cure taking so much longer. In comparison, just quickly with COVID mutates a lot less than HIV, and so there's less diversity, and so we are able to target pretty much a uniform population of viruses when targeting COVID, which is why it was so much easier to develop a treatment that was, not like a vaccine that's effective at preventing transmission of COVID, because the viruses that are being transmitted are so similar to each other, as opposed to HIV, where the diversity of transmitted viruses is very high.
Okay, and then now you talked about like when it comes to infections that HIV is very smart, and when it comes to infecting the immune system. So please just explain how this HIV evades the detection of the immune system. Like, how is it able to infect by being like, how is it so smart? Like, how does it work?
Right. So it's got a number of, we call it strategies, to evade the immune system. So the first of these strategies is actually exactly what we talked about, is the mutations. Because our immune systems are very specific, they recognize what we in the science field called epitopes, right? And these epitopes are just little signatures that make the virus what it is. So in the same way that somebody can recognize you by your face, by your eyebrows, by your nose, that's how somebody else will recognize you. That's how the immune system recognizes the virus. It's got very specific features that identify it as HIV. And as HIV mutates, these features can change slightly. And so that changes the recognition that our immune system has of HIV. This is why mutations and HIV diversity is such a big problem. It allows evasion of the immune system, like non-recognition if it's changed enough. Another way that it does that is HIV's got encodes for these things called accessory proteins, and these basically can bind to some of the proteins in our body that are responsible for recognizing it, and it basically destroys those proteins. And so those proteins cannot function to recognize HIV anymore. So in some ways, in a lot of ways, HIV is very sneaky. So it's sneaky in that it kind of disguises itself with all of these mutations, but also it takes out any of the proteins that can readily recognize it and eliminate it before it can establish infection. These are just two of the ways that it evades detection by the immune system.
The question now that comes up is to say that, okay, we know that HIV is very smart, and with it being so smart, why can't we then be able to be smart also and use or do similar vaccines? Like, we know that when we're young, we get vaccinated against maybe polio or something for a lifetime. Why can't we have a similar vaccine like that when it comes to HIV, to say that when I'm like a couple of months or maybe I'm 5 years old, I get vaccinated against HIV and I'll never be infected?
Right. So it goes back again of HIV and how it mutates, right? So if we consider polio, and polio changes very little along its replication cycle within one individual, but also between cycles of transmission. And so it's a very uniform virus that is being transmitted. So if we train the body with a vaccine, for instance, and we give the vaccine is a little piece of the virus that your body can learn to recognize beforehand. That's how vaccines work, right? So you give the body this little piece of the virus and you say, the body sees it as non-self, develops a response, and then when it encounters the actual virus, it recognizes it immediately and can neutralize the virus before it takes hold, right? With HIV, because it's so diverse and it's so different from person to person, across subtypes, and even within an individual, blocking all those different kinds of viruses with one vaccine is very difficult because you have to train the body with a piece of that virus, but which piece do we use if they all look so different to each other? And so that's, that's where HIV vaccines have not been successful, is that they are not able to train your body to recognize the vast diversity of HIV that is out there that you could potentially encounter,
Okay.
upon exposure.
Oh, wow. Okay. So it looks like this virus is it looks like it's just too smart, but we know that we have smarter scientists and like you and researchers like you, and we know that you've also found ways or strategies that we can use when it comes to working towards an HIV cure. And one of those strategies is the shock and kill strategy. So please explain how it works and how does it aim to eliminate HIV reservoirs?
Okay, so to start this, just quickly, I think I have to go into a third way of how HIV avoids detection by the immune system, right?
Okay.
And I alluded to it in episode 1, where HIV takes advantage of the fact that it is infecting immune cells. So CD4 T cells make up part of the immune system. So essentially HIV is hiding in your own immune system, knowing that your body cannot get rid of your immune system because it's the thing that's protecting you from everything out there, right? And so it can kind of shield itself in these cells and make sure that your body doesn't recognize these cells as having HIV in them. How we try to exploit this in potential cure strategies is why we have the term shock and kill. And so one proposed strategy was, if we can find a way to shock HIV out of hiding in these cells, then either our immune system on its own or through vaccination can now identify these cells that were hiding, right? And so if we shock HIV into producing a couple of these proteins that the cells can raise as flags on the outside to say, hey, look, I've got HIV in me, then we can move on to the kill part of the strategy and actually kill those cells that harbor HIV. And so that's where shock and kill come. You refer to it as a strategy, but there are many ways that we can shock HIV out of hiding. Many potential ways. And that is the strategy to eliminate the reservoir, basically shock HIV out of hiding and then kill the cells that are infected.
Okay, and then we know that, or I know that, let me not say we, because this is a me thing. I know that
Right.
you have also found another approach, and it is called the block and lock strategy when it comes to also working towards an HIV cure. So how does the block and lock work, and how is it different from the shock and kill strategy?
Right. So block and lock is essentially the opposite kind of shock and kill. And so if we can't reactivate all of the cells that contain latent HIV, shock HIV out of hiding and kill them, then maybe we can do the opposite. So we block HIV and lock it in that cell so tight that it's not able to reactivate at all. And so there are these two, I wouldn't say opposing strategies, but these two slightly, well, quite different strategies into how to get rid of HIV. So the first one is just eliminating it, and the second one is blocking it down so far that even if somebody comes off of treatment that suppresses their virus is not going to reactivate and result in viral load rebound because it's locked up so tight. And so, yeah, two different strategies. We'll see how that pans out.
Alrighty. So now that we know about the block and lock strategy as well as the shock and kill strategies, are there any success stories when it comes to using these strategies or any promising results from the studies that are ongoing across the world?
Well, for shock and kill, it has unfortunately not been as successful as we have expected it to be. There are there's a field of cure research that's working on what is called latency reversing agents. And so these are agents that are able to shock HIV out of hiding. And in its infancy, the research was not very effective. So we saw some effects when we were just looking at cells and adding these agents to cells, but in early clinical trials, they weren't effective at. So we were able to shock HIV out of hiding in people living with HIV, but not able to effectively kill the virus or reduce any kind of HIV DNA or HIV burden in those individuals. For lock and block, there's been some interesting evidence from people with HIV who are controllers. So people who are able to naturally control the virus in their bodies to below detectable levels or very close to undetectable levels without being on ARVs. And so their bodies have naturally just found a way to block and lock the virus. And so this could either mean that the virus is integrated in parts of their DNA that's not very easily accessible, so that's very tightly wound, and the HIV cannot be expressed. Or their immune systems are able to very effectively block HIV replication. And so from block and lock, we've seen some good evidence from just from people living with HIV for block and lock. As for treatments that are currently being tested in the lab, I think it's still too early to say whether block and lock would be very effective. Remember that cure research, at least with the on the actual intervention side of things, is still very much in its infancy, in my opinion.
Okay, thank you for that. And then also earlier on, you mentioned that when it comes to the effectiveness of ARVs is because, right, it's a combination of them. You don't just take one particular, it's a combination of them. So then why do researchers believe that a combination of these HIV cure strategies are needed or that are necessary when it comes to having an HIV cure or a functional HIV cure?
Right. So there's a number of reasons for this. First and foremost, I think it's important, again, I mentioned choice, because it's important to have a choice for what kind of treatment individuals want to be on, for what kind of treatment will work for individuals. The second thing is HIV is so diverse that we cannot expect a cure to be one size fits all. That's not to say that each individual needs to have their own individualized cure approach, but it could mean that certain things may work better for certain people and other things will work better for other people, as is with everything in life, even ARVs. We should have different combinations for different cases, and we should focus on what's going to be the most effective for a given individual and or a group of individuals. And so one thing in the cure research field is to try and understand the reservoir so that we can group people into reservoir attributes, for instance. So let's say, okay, this group of people, their reservoir looks like X, Y, and Z, and so these treatments are likely, or these interventions are likely to be more effective within these individuals compared to others where a different strategy might need to be used. And so I think if we are to achieve any kind of successful cure, this would be the best approach to do that.
We've been having such a great conversation, and unfortunately, time is just not on our side. And I just want us to close it off. And with that, please just explain how understanding the science of HIV reservoirs as well as latency helps in the search for an HIV cure, just to close it off.
Right. So, I mean, HIV is a very complicated virus, and if we study it in the context of our bodies, that complicates things even more because we are complicated organisms. And so, understanding the science of HIV reservoirs and how latency forms, how reservoirs change over time within individuals and across individuals, is going to allow us to pinpoint where we would be most effective. I think rushing into anything where we don't understand the scope of what we're dealing with is not a good approach to take in this case because it is so complex. It's such a complex situation. And so, yeah, like hopefully we can continue finding out more about the reservoir, and one day that will lead to us having a successful cure intervention or multiple cure interventions, hopefully.
Yes, thank you so much, because we really want to see some interventions made. Well, we want to see the functional ones, because you are working very hard in terms of research and doing the work to see what is working and what is not. And we just want to appreciate the work that you alongside other scientists across the world are doing when it comes to working towards accessible HIV cure. And thank you for making time to spend with us and just to educate and inform us using the simple language to understand what exactly is happening.
Thank you. Thank you for having me. And it's, like I say, these conversations are very important, and the work that you do is super important. And yeah, hopefully we can keep working together to realize this, dream of an HIV cure.
Definitely. And yes, we will have you back again, because you are now our friend in HIV cure.
Thank you.
All right. Thank you so much. So thank you for tuning in to today's conversation on HIV cure, the build up. And today we're just talking about the science of HIV cure research. We just wanted to find out what challenges they're facing and what interventions are working so far. So thank you for tuning in. I will catch you on the next one. Bye.